Hello Pioneers, Dr. M. I'm here today to talk to you about the use and care of the Olympus CH2 compound microscope. Okay, this is going to be one of your main tools as a biologist, so you want to know how to use and take care of it. And we have some very good scopes for you. The Olympus CH2 has a 10x ocular. It has a rotating turret with 3 separate oculars on it. It has a completely adjustable substage. It has a quartz halogen light with a rheostat that allows it to be infinitely adjustable, as well as the traditional iris diaphragm. Yes, folks, this is one magnificent magnifying machine. Okay, and if you use it and take care of it well, it will last you and last us for a long time. In fact, as it already has. You might want to take a look at your lab manual, the diagram of the microscope on it. I'm going to go through the various parts. As already mentioned, at the top we have an ocular. The ocular will fall out very easily, so you want to be careful. If you look at it very carefully, you can see on it that it is marked 10x. Okay, 10x means that it magnifies by a factor of 10. That will become important in a little bit. There is the head part of the microscope here. There's an adjustment knob on the side. If you loosen that, this can be rotated around. Often your instructor will come to you. You'll want to ask the instructor about what you're seeing. The instructor will loosen that, rotate it so that they can look through, and then rotate it back. Okay? Normally we try to tighten that back up, but double check on us, make sure we don't. It doesn't have to be super tight. You don't want to really bear down on it, but if it's too loose, this whole assembly can come off. So you want to be careful about that. Okay, we have the arm of the microscope. That's where you pick it up. As mentioned, we have the rotating turret. There are 3 separate objective lenses on the turret. This would be B in the diagram. One of them is marked 4x. On most of the microscopes, it has a red line on it. Another is marked 10x. On most of the microscopes, it has a yellow line on it. And the last one is 40x. And it is marked with a blue line. To calculate magnification on a microscope, you take the number on the ocular and you multiply it by the number down here. So for this microscope, the 4x objective times 10 would give us 40x. The 10x objective times 10 would give us 100x. And the 40x objective times the 10x on the ocular would give us 400x. That's the That is the most magnification you can get with this scope. There is another port on here for an oil immersion lens, and in our microbiology lab we have better microscopes, and they have the oil immersion lens on them. Okay, as mentioned, we have a stage below here. Now these microscopes are a little bit unusual. Not all microscopes do this. Some work a little bit differently, so you want to pay attention. Some microscopes, when you adjust the focus, it moves the objectives. Most are like ours and move the stage. But I just wanted to call that to your attention. If you're used to one that moves the objectives and you're turning the focus knob waiting for something to happen, you might be looking at the wrong part. The focus knobs, there are 2 of them. There's coarse adjustment, that's the big knob down here, and fine adjustment, which makes very small movements. Okay, you should never move the stage upward unless you're looking at the side. Never move the stage up while you're looking through the objective. I'll explain why in just a minute. Okay, C on this diagram is supposed to be referring to this whole stage. It looks like it's pointing just to the center, and that is the top of the condenser assembly that we have down here. The condenser assembly has some light lenses that focus the light. And there is also this lever here, which is marked D. That's the iris diaphragm. And that makes a major difference in controlling the amount of light that comes through. Generally, you should have this condenser assembly moved all the way up. Okay. Letter E refers to the lamp down here. And letter F refers to the knob, which is on the other side of the microscope. You couldn't see me adjusting that. That is located right here. Okay. Let's see, what else? We have G and H. Again, I pointed those out. G and H. G refers to the coarse focus. H I'm going to come back over here, refers to the rheostat. That's R-H-E-O-S-T-A-T. That's the fancy name for the electric control for the light that controls the amount of light. Two ways to control the light. One is with the rheostat. That will also change the color of the light. Generally, you want it turned up a bit, 7, 8, 9 or higher, and then do the rest of the adjustment with the iris diaphragm. The iris diaphragm, in addition to controlling the amount of light, controls the clarity of what you're seeing. Just like when you squint your eyes down, if you really want to see something, moving the iris diaphragm will allow you to see a little bit more depth on your slide. So you may find yourself adjusting it for that purpose as well. And finally, I is referring to the fine adjustment on the microscope. So those are the basic parts of the microscope. A few words about the care once you have the microscope out. We will be cleaning the oculars before you come in. The other parts of the microscope we won't be cleaning very carefully. And so you might want to spray off one of the self-care towels that's in the lab, get a little, just barely dampen it, and wipe off the parts of the microscope. Nothing that's glass, just the parts you touch, the knobs and so forth, to make sure that it's clean before you use it. The chance of picking up COVID from a surface is very low, but it is real, so you do want to do that. And of course, you've washed your hands when you come into lab, and again, wash your hands periodically during the lab, just to make sure you're not picking anything up, okay? The exception to what I've been just saying is the ocular. You don't want to put your eye against the ocular. It has enough relief that somebody wearing eyeglasses should be able to see clearly, so you don't have to press your eye up against it. But if you would come into contact with your eye, that's potentially a place where you could pick something up. So we will be cleaning these off beforehand using a little bit of standard eyeglass cleaner and a lens paper. You just give that a little spritz, and I'll be clearing mostly the ring that goes around the outside of this. But if the eyepiece looks dirty, we will go in there and clean it as well. You will not be cleaning anything on the microscope. If it looks dirty to you, make sure that your assistant or instructor knows, and they will clean it, because we use lens paper to clean off the oculars and the objectives, and these Kim wipes here can be used for other things. Now, the one exception to what I said about cleaning, like I said, in addition to maybe wiping it down, if you spill water on the stage, you can wipe it off with one of the Kim wipes. Just avoid wiping on the glass surfaces. Okay, but these will be, this part will be cleaned before you come in. The rest of it, just give a gentle wiping down with the disinfectant spray provided in the lab. Okay, so that's the basic use of the microscope. And I want to talk now a little bit about use in practice. Of course, normally I'd have the microscope plugged in. I don't have an outlet up here, so I won't do that right this moment. Again, we put the condenser all the way up. Once we've put our microscope slide on the stage, you look from the side and you bring the stage all the way up, making sure you don't crash it into one of the lenses. Can't do it with the small objective lens, but with the big one, that's a possibility. Then you look through the microscope and focus downward until something comes into focus. Okay? Once you have it in focus at low power, you should be able to rotate the next objective in, and it will be very close, that you just need to touch up a little bit with the fine focus. This is a feature that's called parfocal, and these microscopes have it. It's not perfect. Likewise, when you have things adjusted at the medium magnification, you can bring in the high magnification. Again, look to the side, make sure you're not going to hit anything, and then only focus down. If you do find that you have to come up, look from the side, make sure nothing is going to hit, move it up as far as you can without touching the slide, and then focus downward. Okay, so that covers this. One thing you'll notice is that when you switch to the higher powers, they let less light through. You may need to adjust your iris diaphragm. Also on these microscopes, particularly at low power, if you see nothing at all, no matter where you focus, what is probably happening is you have the rheostat turned up too high, and you have the iris diaphragm all the way open. All you're seeing is bright light. Try turning down the iris diaphragm until it starts to get dim and then focus, and that may make a difference. When you go to higher powers, you may need to let a little bit more light in. Okay? So that's the basic use of the microscope. Okay, finally, you're wondering, where do I get these great microscopes from? Okay, they're right here in the lab room. They're usually in the gray cabinets. It depends on which room you're in. Gray cabinets here. Little trick on these. One of these handles is absolutely fixed. And you'll pull on this, and you'll pull on this, and you'll say, Dr. M, the cabinet is locked. Okay, little trick here. The one handle will rotate like that, and then you can open it up. Okay, so you open it up, and you choose your microscope. You want to grab it with both hands. Okay, cradle it in. Those of you who are football players, you know what the coach is going to say if they catch you with the football in one hand. Okay, the reason we want you holding it with both hands is not because we think you're a klutz, but because when you're holding it with one hand, it's easy to come around a corner and slam it into something. We're reasonably confident that you don't walk into things. So hold it with both hands until you get back to your seat. Okay, once you're back at your seat, you can set the microscope down, pull the cover off, and return the cover to the cabinet. You might want to set the microscope down on a nearby table, take the cover off there, and put it back in the cabinet before you carry the microscope back to your seat. Okay, so just be a little bit careful with the microscopes. When you're done, you reverse the process, get a cover, and find an open spot. Place it in the cabinet, try not to bang it like I just did, and when everybody's done, we close the cabinet up. Okay, so that is putting the microscope away. And the absolute final thing I want to mention is we do have some microscopes like this out. These are dissecting microscopes. They work on reflected light, not transmitted light like the compound scopes. Don't get one of these out unless you're instructed to. Normally we'll have these out for display purposes. We might ask you to get them out on certain occasions. Usually when we're telling you to get a microscope, it's the other type, the compound microscope that we want you working from. Okay, but these microscopes are in the cabinets. Don't get one of these out by mistake. Okay, that does it for the microscope lab, for the intro to it. So hopefully you're all ready when you come in to lab. You'll be all set to use the microscope. Hey Pioneers, Dr. M again. This part of the video I want to show you how to make slides and what you might see when you look through the microscope. So I've got the things I need set up here. I've got my test tube rack with my sample, have a pipette to transfer it, have the microscope slides, I've got the cover slips, have some prod slow in case anything is moving fast, and some Kim wipes in case things get messy. So when I go to make a slide here, what I'm going to do is I'm going to take my microscope slide. I'm going to give it a wipe with the Kim wipe if it's not clean. I want to make sure that there's no finger thumbprints on it because that will give us an artifact. I set the slide down, and first thing I'm going to do is put my sample on there. Use a little micro technique here. Grab the lid with my hand so I'm not setting it down. Go in here. You generally want to get a little bit of stuff up off the bottom and put a good sized drop, not a huge drop, maybe 1/4 of an inch across on your slide. Close things back up and set them aside. Okay? Now, if you have animals in your sample, if you're working with allergy or something like that, you might want to put a drop of this Protoslow on there. And generally what you do is you set it right down next to the other drop there, and let them merge together. Okay, then I'm going to take a cover slip and make sure it's clean. It's difficult to wipe these off. Got a little air bubble there, so I'm going to try to pop it. It's not going to pop. So I'm going to set this in. I'm going to bring it in at a 45-degree angle, and I'm going to touch it to the edge of the drop. It will flow across the cover slip, and then I lower the cover slip down and let it go. And I've got my slide made. Okay, so this is ready to go. Spilled a little Protoslow there, so I'll wipe that up. And my slide is really kind of bounced around. I got a little bit extra fluid on there. I'm just going to touch the Kim wipe down, soak up a little bit of that water. Okay folks, I got the slide made, got it on the microscope. I've looked from the side, I've adjusted up to the furthest point. I'm all ready to go, so I'm going to go ahead and take a look, record to the video card so you can see what I'm seeing. Let's take a look. Whoa, that ain't right. Okay folks, here we've got something under the microscope. It's round, it's very, again, very thick cell wall. Moving around a little bit there, let's take a closer look. At higher magnification, you can really see that cell wall, just a hint of structure inside there, kind of jiggling about. Kind of interesting. We'll even want to take a higher magnification look at that. Bring it into focus, and oh, there's a little diatom there in yellow underneath it. There we go, though. You can see the internal structure of the cell now, the nucleolus, some of the other. Folks, this is an air bubble. Okay, sorry about that. You're going to see air bubbles, though. Another interesting thing you'll see is that edge of the cover slip over there to the left. And off to the right a little bit, if we move over and we focus carefully, we'll see something extremely interesting, and many of you will come across. It's a piece of lint, a little bit of Kim wipe that got torn up. Okay, you'll see that stuff. Here we've got a whole family of small organisms, various size. You can see that, no, these are air bubbles again. Kind of get the feeling those round, hard circles. Take a look at something else here. We can see production taking place. So that upper cell is blebbing off, and then below you have a bud that's air bubbles again. Get used to recognizing air bubbles, okay? Round with a black wall. Okay, this is the real stuff now. These are some small algal cells. There's a little ciliate swimming around in there, moving them around. We're going to focus in a little bit more on that big clump right there. And if we do that, we can see that this is something, the technical term for this is called crud. Actually, it's called detritus. This material is dead, decaying organic material. It's been colonized by a bunch of microorganisms. There's algae in there, there are fungi, there's little worms, there are protozoans you can see swimming around in there, a whole host of things. You can see some fungal filaments there. Oh, and here we go. These are really neat. This is something called Vorticella. Okay, it's a protozoan. They have a little ring of cilia. They have a long stalk. They can pull themselves back. But Vorticella, okay, a neat little protist that hopefully you'll see in lab this week. There you can see that, those cilia bringing stuff in. Another close view, you can see how they bring the food right to them. So we're looking forward at seeing you in lab this week. Hope you're all ready to go, and hopefully you'll see some really great stuff besides air bubbles in the microscope lab this week.